Okay, so we're going to continue talking about the structure of nucleic acids. And in this part, we're going to talk about how the nucleic acids, your DNA in particular, is going to be packaged into your nucleus so that it fits. Some important proteins in this packaging are going to be our histones. And so we'll discuss our histones and their functions. And then we're going to talk about the difference between heterochromatin and euchromatin and how that impacts epigenetics of different cells. So let's talk about histones. So first off, what we're talking about here are our chromosomes. And so our chromosomes are going to form structures that are called chromatin. And chromatin is really the structure of the DNA. And the chromatin are going to depend on our histone proteins as well as our non-histone proteins that are going to be important, such as our polymerases, transcription factors, and other proteins. Now our histones are going to form a core, and this blue string is the DNA wrapping around the core. Some things to note about the histone proteins is that they are basic. The DNA is negatively charged, and so they are going to be attracted to each other. And the histone's purpose is to package the DNA into nucleosomes. And these nucleosomes are then going to be able to fit or be compacted in order for the DNA to fit inside of our nucleus. Now, when we talk about the actual histone proteins, there are a few things to note. One is that you have a high amount of arginine and lysine. Again, remember, they're going to be basic proteins. And that arginine and lysine are going to make about a quarter of these histone proteins. There are 5 histone proteins. 4 are going to form the core. So these 4 form the core. Histone 2A, 2B, 3, and 4. And histone 1 is going to be the linker. So we're going to have histone 1 link the different nucleosomes together. Now, histone 3 and 4 in eukaryotes are almost identical. So this means that they're highly conserved across different kingdoms within the eukaryotes. Now, what's the purpose of all of this? Well, it's to package the DNA. The DNA is too large to fit inside of our nucleus. Even in bacteria, their DNA is too large to fit inside of their own cell. And so these cells have come up with a mechanism to package the DNA, and we use our nucleosomes. Nucleosomes are going to form the core, which is a higher packaging of chromosomes, and that core is going to be made up of those 4 proteins doubled. So each protein in the core is going to be found twice. So you're going to have 2 of your histone 2A, 2 histone 2B, 2 histone 3, and 2 histone 4. So you need 8 proteins to make up that nucleosome core. And then, as I mentioned before, histone 1 is excluded from the core, and histone 1 is a linker histone protein. Now, the nucleosomes are going to form a string, and then they're going to wind into coils, and these coil structures shown here are the solenoid structures. And solenoid structures are therefore going to be able to compact the DNA. Now, the human genome is going to be made up of 46 chromosomes. You're going to have 23 chromosomes that are going to have a pair, so that equals 46. 22 of these pairs are autosomal, and these are going to be common throughout all of us. And then there are going to be 2 sex chromosomes, and females are XX and males are XY. So genetic males are XY. And so all of these chromosomes contain our genetic information, and they're all going to be compacted into our nucleus using those solenoid structures. Now, within these structures, there are 2 different regions. There's heterochromatin and euchromatin. Heterochromatin is going to be more densely packed, so it's going to be those darker staining regions. So if you look up here, we have darker staining and lighter staining regions. And then the euchromatin are going to be loosely packed, and they're going to stain lighter. The loosely packed regions are going to be where you have active gene expression, So transcription is going to be taking place. So it's this area that will open up, allow your RNA polymerase to come in, make a messenger RNA, and then eventually that will become a protein. Your heterochromatin is going to be more densely packed. You're going to have these densely packed areas near the centromere because centromeres do not encode genes. And these are areas of transcriptional silence. Now, how does a cell know which areas should be expressed and made into proteins and which areas should be silenced? And this is going to be an epigenetic process. And epigenetics is important to understand because epigenetics is basically going to tell you your cell, you're a hepatocyte, so you need to make liver proteins, or you're a neuron and you need to make neuron proteins, or you're a kidney, you need to make kidney proteins, etc. So every cell has the ability to make any protein. It's really the epigenetic process that tells the cell what to do. And these epigenetic processes have to be universal, so they use universal mechanisms in order to make certain genes available in a hepatocyte that would be turned off, for example, in your neuron. And it's really straightforward. So methylation is going to be used to modify your DNA. So methylation of cytidine residues, or CpG, dinucleotides, is going to be a process to silence genes. So epigenetic processes are going to alter the chromatin structure. And again, it's by making some areas more densely packed so they're not going to be transcribed and translated into proteins. And other areas are going to be more loosely packed. And they're going to affect the activity of gene because they're going to allow hepatocytes to only express liver proteins, and they're going to allow neurons to only express neuron proteins. So epigenetics can change the phenotype by controlling gene expression. It does not change DNA sequences. Mutations, on the other hand, are going to change DNA sequences, and mutations are not epigenetic processes. So histones can be modified as well. So our histone proteins are going to be acetylated in areas where you're going to have expression of genes. And they're going to be methylated in areas where you're going to have silencing. And so again, methylation is going to be a process to silence genes, an epigenetic process to silence genes. And what's important to note is that methylation has to be transferred to daughter cells. So if a hepatocyte divides, each daughter cell will not only get all of the information in their chromosomes, but each daughter cell will also have an imprint of methylation that will be passed on so that that hepatocyte daughter cell becomes a hepatocyte and not a neuron, for example. So methylation, again, is going to be on the 5-carbon of cytidine. And then this is also known as CpG motifs. Your methylation is going to be about 60 to 80% of all cytidines in our chromosomes are methylated. And again, this is to silence those areas. We have 3 genes that are going to encode our methyltransferases. We have methyltransferase 1, 3A, and 3B. All of these are going to use SAM as a single carbon donor, SAM as adenylyl methionine. So this is going to be dependent on vitamin B12, folic acid, and methionine. So as we can see here, vitamin B12 deficiency, folic acid deficiency are very serious because we use them in all our cells for epigenetic purposes as well. So during cell division, again, these patterns of methylation have to be replicated. And so you can see here we have cytosine, and that's going to get methylated at that 5-carbon position. So it's a 5-methyl cytosine. And then this can get deaminated. So in this case, we're removing our nitrogen, and it can become thiamine through deamination processes. Now, there are some other histone modifications to note. So histone modifications are going to mostly occur on 2A and 2B. Remember, 3 and 4 is going to be shared across many of the different mammalian species or eukaryotic species. 2A and 2B are going to be more unique to humans. And so 2A is going to have ubiquination. Ubiquination is a protein, ubiquitin. So this is a protein modification. And then adding ubiquitin to 2A is going to suppress or repress transcription. Ubiquitation, excuse me, to 2B is going to stimulate gene activity. So it's just changing the structure of your proteins that are compacting your chromosomes into your nucleus, and changing those structures allow regions to open up and be transcribed or closes them. Histones can also be phosphorylation, and this phosphorylation is similar to what happens when we have signal transduction pathways, in that it is a quick way to activate things. And so phosphorylation is going to occur following things like stress. And then you're going to enable cells that are going to be responding to stress to be active or become available. So in summary, we have an organization of our nucleic acid, our DNA, in the form of chromatin, the structure of DNA. And we're going to have the chromatin is going to be formed into nucleosomes with a histone core and a histone 1 linker. Nucleosomes are going to fold into solenoid structures. The nuclear core is going to be made up of 4 proteins that are in duplicate in every core. And each of the cores are going to have about 146 base pairs that wrap themselves around them into that solenoid structure. And then the nucleosomes are going to be linked by that histone 1. Chromatin structure is going to depend on epigenetic processes, methylation, ubiquitination, phosphorylation, acetylation. Those type of things are going to help to control. And remember that epigenetics is not mutations. It is simply controlling what genes are available for expression and what genes are going to be silenced.